I'd like to thank Dr. Douglas for the opportunity to speak here today. And we're developing the world's most effective oxygen therapeutic for treatment of a number of life-threatening conditions. And we have a collaboration with Dr. Bill Culp at the University of Arkansas, and he's done pioneering work in stroke with our product. The technology relates to the discovery that microbubbles transport far more oxygen than any other material that's ever been studied, and the discovery that fluorocarbons carry large amounts of oxygen, and then the development of fluorocarbon microbubbles to carry oxygen. So we're applying this to a number of life-threatening conditions where oxygen is needed to keep tissue alive. So if we think of stroke, hemorrhage, other conditions, oxygen delivery is compromised, and so this could be useful for delivery to a variety of life-threatening conditions, and could be applied early in the course of care to treat stroke, heart attack, severe blood loss, hemorrhagic shock. And in cancer, radiation therapy is used to treat many patients. And the mechanism of radiation therapy is that it acts with the oxygen to strip molecular oxygen apart to create singlet oxygen that then destroys the cancer. And so if a tumor is hypoxic, it doesn't have enough oxygen for the radiation to work. So we have strong data in stroke, hemorrhagic shock, and cancer showing that our material is far more effective than anything that's been previously discovered. Here is an in vitro study showing the oxygen absorption of our material at 37 degrees centigrade body temperature and at room temperature compared to saline or to the liquid fluorocarbons, perfluorodecaline or perfluorooctobromide. Billions of dollars were spent on the clinical trials for perfluorooctobromide, over $1 billion, testing it in various clinical conditions. And our material, as we'll show later, uses less than 1/100th of the dose. So the liquid fluorocarbons ultimately failed due to the high doses and adverse events, and our material requires less than 1/100th of the dose. This is work that was done in collaboration with the Department of Defense with our material. Rats were bled out, in this case removing essentially all of their blood volume. So the rats start with a hemoglobin of about 16. They go down to less than 2, and all of the control animals die when the hematocrit is below a level of, say, 6. And these are two groups of animals here receiving our material, and they all survive up to 240 minutes, giving a single dose of 0.7 cc's per kilogram. That would translate to a human about 50 cc's, so about 50 cc's of our material is enough to replace the oxygen carrying capacity of the entire blood volume in a human. This is again with the Department of Defense, and this is hemorrhagic shock in a pig model, and they were interested in testing the material in a battlefield type scenario. So the pigs had blood loss of greater than 50% of their blood volume, and also had a fracture of the femur to mimic hemorrhage polytrauma. And in the control animals that received a standard plasma expander, all of the animals die by 180 minutes. What we're looking at here is carbon dioxide production and oxygen consumption, both of which fall off as the animals go into shock and die. Giving a dose of 0.7 cc's per kilogram of our material to the pigs, they stay alive, they have normal oxygen and CO2 production, out here at 400 minutes, and the animals were given fluid and then allowed to recover, and at 30 days then were killed and had normal organs and so on, so no sign of the sequela of shock that would occur otherwise. Here is the pioneering work that Dr. Culp is doing at the University of Arkansas in ischemic stroke, this is a angiogram in a rabbit, the normal angiogram, and he has created here an embolic stroke, an occlusion of the middle cerebral artery. And rabbits are a very good model because there is only one drug that is approved by the FDA for treating stroke. It's called tissue plasminogen activator, and it was studied in rabbits. So rabbits are a very relevant model for stroke. And so Dr. Culp creates a stroke, and he's also able to look for hemorrhage in the stroke. And we find that our material decreases the brain damage by about 90% compared to the controls, with no increase in hemorrhage. And so here are the control animals showing the volume of the infarct compared to animals receiving our material at different time points following the stroke, up to 3 hours following the stroke. The animals have a dramatic reduction in the size of the stroke, and we've studied stroke out to 7 hours and see benefits. And preliminarily, some studies have been done to 24 hours where we also see the benefits. So what is the potential market for this technology? We're calculating a dose at $1,000, and if you look at, as I mentioned, TPA, treating stroke, or various mechanical devices used to treat stroke or heart attack, certainly they are more expensive than the $1,000 that we are looking at. But so the U.S. market is over $5 billion. Heart attack is the #1 cause of death. There are 1.5 million heart attacks per year in the United States. Stroke is the #2 cause of death, #1 expense for long-term medical care. There are 700,000 new cases per year, and again, dramatic reduction in the size of stroke in the rabbits. Cancer is the #3 cause of death, and this is referring to patients treated with radiation, with hypoxic tumors. There are over 1 million patients treated with radiation each year in the United States, and the average number of treatments in those patients is 28 radiation treatments. We're forecasting here that we would have more effective treatments. We'll be able to do 10 radiation treatments, and so that's 3 million treatments per year. And there are over 100,000 cases of hemorrhagic shock per year. So our product should improve survival and decrease long-term disability. There are no effective competing oxygen therapeutics. A lot of money was invested in the liquid fluorocarbons, and they required high doses, more than 100 times the dose of our agent, and they had adverse side effects. So we have a small dose with no adverse side effects, and this material that we're using was previously tested in over 2,000 patients as an ultrasound contrast agent, and it was found to be safe, and we have right of reference to these documents, and we have licensed 4 U.S. patents, and our own company has filed soon to be 2 patents, and we are licensing a patent from the University of Arkansas, in which I am a co-inventor with Dr. Culp. NVX-108, we've been discussing this with the FDA, and NVX-108, our product, will be regulated as a first-in-class biologic. Now, this is very important because biologics are granted 12 years of exclusivity in the marketplace for an approved indication. And so this doesn't matter whether you have any patents at all. If you're the first-in-class biologic, then you get that exclusivity. So just looking here at the dose that I've been talking about, published studies in stroke, here's the dose of our material, about 28 milligrams per kilogram, and the other liquid fluorocarbons over 6 grams per kilogram. So that's why we think we're going to be successful, is our material is just so much more active because of the surprising discovery that microbubbles are more effective in oxygen delivery. Again, no competing oxygen therapeutics, and we are poised to become the world's first effective oxygen therapeutic to address critical life-threatening conditions. We are summarizing the regulatory studies that we have in our possession, and probably will be finished doing that next week. And then either at the end of this month or in November, we should submit our pre-IND to the FDA. And our proposal will be that we go into human studies in stroke patients rather than to repeat safety studies. And so, of course, we will need their input into that. And then the remaining clinical trials will take from 3 to 5 years to complete. Development team, I am the president CEO. I'm a radiologist, and I have developed in the past 3 FDA-approved drugs and 1 FDA-approved medical device. And Definity is one of the products I developed. It's the #1 selling ultrasound contrast agent in the world. It is a fluorocarbon microbubble, and it sells over 125 million in this country. And I'm an inventor on over 100 issued U.S. patents. Jennifer Johnson, who is attending the conference with me, is our Chief Operating Officer, and she has extensive experience in drug formulations and emulsion technology. Terry Matsunaga is our Chief Scientific Officer, and the 3 of us are co-founders of NuvOx. We have a Chief Financial Officer and several scientists working for our company. We have a number of external collaborators, including Dr. William Culp, and collaborations with several different groups in the Department of Defense and in academia, radiation biologists and cardiologists. To date, we've received $1.5 million in grants from the National Institute of Health. We currently have over $1 million in grants that have been applied for that are pending. The 3 founders have invested more than $500,000 in the company of their own money. We would like to raise $6 million, which would enable us to complete the phase 2 clinical trials in the stroke indication. And if you're interested, we'd love to talk to you more about our project. We're very excited about this technology to make a difference in these serious diseases. Thank you.
Questions for Dr. Unger?
Have you looked at applications, prophylactic type applications for this, such as maybe military, and how the effect of it delivered at different spots in time, pre and far post the event?
The military is interested in nitrogen narcosis, because they're concerned if, like, a submarine goes down and they have to rescue the people. And we do have data that we increase the washout rate of nitrogen in nitrogen narcosis, but we haven't looked at prophylactic applications.
Dr. Unger, over here, you indicated, I may have missed something, that you were going to either petition the FDA, the FDA to go into stroke victims rather than conduct a safety study. Could you talk about that a little bit more?
There were 2,200 patients that were given this material in clinical trials for as a contrast agent, and they did a safety study, and there were 91 animal studies that were performed with this material, 35 to 40 of which we have complete reports. And so we've summarized all this data, and our intention is to present the data to the FDA and request that we could then go into a clinical trial in stroke patients. And so we have to see if they agree with that, but we have right of reference to those documents. We have the exclusive, we own those documents. Mm-hmm.
Are the levels used as of this compound as a contrast agent greater than the levels that are being used here for therapeutic use?
In the animal studies, there were repeated administrations of doses higher than anticipated for the stroke application. There were some human studies administered similar doses. Okay. And one of the differences was that in all of those studies, the material was administered as a rapid IV bolus. For stroke and all the applications that we're talking about, we're envisioning giving an IV infusion typically over, say, 30 minutes, and that should increase the safety factor.
Thank you. Other questions? Okay, Dr. Unger, thank you very much for your presentation.